Hello, everybody. It's Mr. Cadell. I hope you're doing well. I'm doing well. Welcome to biology second semester. I hope that you have a good time. I hope that you learn a lot about biology, and I hope that you have an open mind and that you can see how wonderful the complexity of life is and all the details that go into that, and just let it wash over you and be accepting of information, but also question everything for sure, and try to make me better, make yourself better, and learn as much as you can. I'll tell you one big difference between this and the first semester is that we're trying to get out of the cell here. I know the first semester had to do with all these little tiny things that you couldn't see. A lot of them were kind of just outlined as cartoons in the book, and some kids really find that hard to make all that stuff tangible. So this semester we're going to not only spend 2 of the 4 units on ecology, which is basically the science of organisms in their environments. So that means we're going to go outside and look at the hillside and go to the lake and try to really get moving outside. But we'll also do evolution, which is my personal favorite concept in all of biology. One of the reasons why is because it puts together all of the themes and concepts that you've learned before, and then there's competition involved. And only the most efficient and best things or characteristics or organisms survive. Right now, we're doing heredity, which some kids really like. They like to see how information is passed from parents to offspring, and I hope that you do too. We have a textbook. It's a pretty good textbook. It has a lot of good content in it, but it doesn't have great activities. We're going to actually use 2 other textbooks for activities, and then I have some of my own personal favorites. I've done this for 20 years, so I just have some things that have worked well for me in the past. For right now, I want you to do 2 things. I want you to get out a blank piece of paper. If you were in class, I would give this to you. And I want you to put your hands together, and I want you to rub them for like 5 seconds. Rub them over the table and see what's there. Can you see the cells that are on the table? They're microscopic. Too small for the naked eye to see. But I'm going to tell you, you probably lost about 1,000 of them. Every time you put on a shirt, every time you go to the bathroom, every time you touch a doorknob, you're losing cells. Now, if you were to be able to look at one of these little individual cells and look at the DNA within the cells, what would you see? How many cells do we have? We have about 50 trillion cells, some scientists estimate. And within each of the cells, they have a nucleus, which has DNA. And if we were to stretch out all the DNA in a single nucleus of a single cell, how long would it span? What's the distance? And the answer is 6 feet. I want you to wrap your head around that for a second. So that means there's about as much DNA as the height of me, 6 feet, in every one of these cells that are microscopic, which means you can't see it with your naked eye. You can see a human female egg that's come out of the ovary. I've seen one on a slide before, but you can see it with your naked eye. And then conversely, the male sperm is the smallest cell that humans create. But the point is that these are super small structures, and they have 6 feet of DNA within them. Now, all that DNA is coiled up into an area that's about 200, or a distance, which is the diameter of the nucleus of the cell, which is about 250,000 times smaller than the actual length of the DNA itself. So it's super wound up, and we're going to get to what that looks like in chromosomes in a few minutes. How many letters of information of these nucleotides exist in each one of these 6 feet of DNA in each cell? The answer is pretty stunning. There's 6 billion letters per cell. What I mean by letters is if we were to write A and T and C and G and A and C and T and A and whatever, as you go down the side of a DNA molecule, you'll see a bunch of these nucleotides. Some of them are important in creation of proteins, and some of them are just nonsense. We don't really know what they do yet. But there's a lot of them, 6 billion per cell. So I want you to wrap your head around that. There's 6 feet of DNA in every microscopic cell, so small that you can't even see it. There's 6 billion of these letters in a very specific sequence in each one of those nucleuses, and then there's 50 trillion cells in your body. So like I said, just be amazed by biology. And I mean, can you wrap your head around those numbers? I certainly can't. I'll tell you, when these letters are wrong, if any of these are wrong, sometimes when some of these are wrong, doesn't mean anything, doesn't change the protein that's created. There's no specific symptom that you see in the organism. But sometimes when these DNA letters are incorrect, you get incorrectly folded proteins or misshapen proteins that don't do a job. And then there is a symptom. Someone is deaf, or someone is blind, or someone can't digest a certain vitamin, or person has a certain hair color or height, or whatever it may be. There are consequences of changes in the nucleotides of the DNA. We're going to start here with a movie.
Every human being starts out the same way. 2 cells, one from each parent, found each other and became one. And that one cell reproduced itself, dividing, until there were 10 trillion. You realize there's more cells in one person's body than there are stars in the Milky Way? But those 10 trillion cells aren't just sitting there in a big pile. That would be for a pretty boring human being. So what is it that says a nose is a nose, and toes is toes? What is it that says this is bone, this is brain, and this is heart? And this is that little thing in the back of your throat you can never remember the name of. Everything that you are or ever will be made of starts as a tiny book of instructions found in each and every cell. Every time your body wants to make something, it goes back to the instruction book, looks it up, and puts it together. So how does one cell hold all that information? Let's get small. I mean really small. Smaller than the tip of a sewing needle. Then we can take a journey inside a single cell to find out what makes up the book of you, your genome. The first thing we see is that the whole genome, all your DNA, is contained inside its own tiny compartment called the nucleus. If we stretched out all the DNA in this one cell into a single thread, it would be over 3 feet long. We've made it fit in a tiny compartment that's 1,000,000 times smaller. If you just bunched up like Christmas lights, it can get messy. We need some organization. First, the long threaded DNA wraps around proteins closer to the little beads called nucleosomes, which end up looking like a long beaded necklace. Then that necklace is wrapped up in its own spiral like an old telephone cord. And those spirals get layered on top of one another to make a neat little shape that fits inside the nucleus. Voila! 3 feet of DNA squeezed into a tiny compartment. Only we could hire DNA to pack our suitcases. Each tiny mass of DNA is called a chromosome. The book of you would have 46 chapters, 1 for each chromosome. 23 chapters of your book came from your mom, 23 chapters came from your dad. 2 of those chapters, called X and Y, determine if you're male, XY, or female, XX. Put them together, we get 2 almost identical, but slightly different sets of 23 chapters. The tiny variations are what makes each person different. It's estimated that all the chapters together hold about 20,000 individual instructions called genes. Written out, all those 20,000 instructions are 30 million letters long. If someone were writing 1 letter per second, it would take almost an entire year to write it once. But it turns out that our genome book is much, much longer than just those 30 million letters, almost 100 times longer. What are all those extra pages for? Well, each set of instructions has a few pages of nonsense inserted and have to be taken out before we end up with something useful. The parts we throw out we call introns. The instructions we keep we call exons. We also have hundreds of pages in between each gene. Some of these excess pages were inserted by nasty little infections in our ancestors, but some of them are actually helpful. They protect the enzymes from being damaged, or some help our cells find a particular thing they're looking for. Or give the cells
Link crew leader, come down to the lobby. Link crew leader to the lobby. Please clean up your station.
For book's 46 chapters is between 48 and 250 million letters long. That's 3.2 billion letters total. To type all that copy, you'd be at it for over 100 years, and the book would be over 600,000 pages long. Every type of cell carries the same book, but each has a set of bookmarks that tell exactly which pages it needs to look up. So a bone cell reads only the set of instructions it needs to become bone. Your brain cells, they read the set that tell them how to become brain. If some cells suddenly decide to start reading other instructions, they can actually change from one type to another. So every little cell in your body is holding on to an amazing book full of the instructions for life. Your nose reads nose pages, your toes read toes pages. And that little thing in the back of your throat, it's got its own pages too. They're under your uvula.
Okay, sorry about that interruption there. You saw some numbers that they threw out in that movie, and they were half of the numbers that I quoted earlier. What's often what people do is they only count one side of the DNA, and I was counting both sides of the double helix earlier. This is what is going to guide us for the next few days in this class, and I'm going to read this. Explain how cells form specialized tissues due to the expression of some genes and not others. You have to understand that the DNA that existed in the very first cell, when your mom and dad got together, I should put my hands away, when they got together and the first cell was created, there was some DNA in that very first cell. That's called a zygote. And then that cell has divided and divided and divided trillions of times until you now have a grown human being. But the point is that in every cell, you have the same exact DNA as in every other cell. And so the point is that some cells are going to turn on certain genes, like the movie just said, like the cells in your nose are going to turn on genes that will create proteins that will create tissues that make cartilage to make your nose. But those same DNA molecules, exactly the same, because they came from the first original cell, just like the cells that exist in your nose area, they are going to, let's say cells that exist in your pancreas, they're not going to turn on the genes that create the cartilage to create nose tissue. They're going to turn on the genes that create pancreatic tissue. And so here's the question that you see. This is the state standard that's put out there by the Colorado Department of Education that should guide all of our instruction for whatever part of the unit that we're on. Why do some cells turn on some genes and express those genes and create those proteins and some don't? So that's the state standard 2.8a. You will have all this on this piece of paper. This piece of paper is important, and it's going to guide you for the next few days. I'd like you to put your name on it, and then you're going to write down the activity that we're about to do, and then you're going to staple all this work to the back of this, and you're going to hand it in to me. So you don't hand in to me like individual assignments unless I ask for them. But in this case, in order to help you gain mastery of this state standard, and then there's some others on the back and stuff, you're going to just keep all the work together. So you've got to get organized in this class. I would say definitely get a binder that's only exclusively used for this class. That would be a big tip I would give to everyone right now. And then just get organized, because you might do all this work and then lose it 3 days later. And then I say, all right, turn it in 6 days after you did it. And then all of a sudden you're like, I don't have it. And that sucks for you. So get organized, please. So look a little bit more at what this looks like. You can see there's just some things for self-reflection there. Then I would like you to write down the different assignments as we do them on these lines. I'm going to flip this over now. You will then see two other state standards at the top there. There's some things, it says students will be able to use their learning to do these things. There's a big idea. There's an essential question there. Just take a moment to take a look at this. I think it's important. You should never wonder why we're doing something in this class. I remember sitting in your seat saying, this teacher sucks, doesn't know what they're doing, they didn't prepare for today. And that was most of college. No, sorry, when I thought I was really all that at the end of college. But the point is I said to myself, hey, if I'm ever in this role, I'm going to be transparent, and I'm going to help kids really understand why we're doing what we're doing. So this is my effort to be transparent. Call me on it. If you're like, this is busy work, why are we doing this? I will absolutely say you don't have to do it. Everyone in class gets an A. Hold me to that. Well, let's talk about genetics. So here's two people. Let's imagine that we consider this lady tall and we consider this guy to be short. I don't know if they are or not. But let's then look at some genes that might be associated with the characteristics of tall or short. So I'd like you to draw this on your piece of paper. Go ahead and draw a Punnett square. You probably remember this from middle school. And then just understand that the baby that they create will have chromosomes from each parent. So let's come over right here. Do you remember how many chromosomes are in every one of your cells? It's 46, right? But that came 23 from dad, and then there's this equal, it's called a homologous set of chromosomes from mom. So here's the sperm, here's the egg, these things come together. Isn't that amazing? I missed the most important one here, the zygote. The zygote is the very first cell that's created when the sperm and the egg get together. And so if you add these two amounts of chromosomes up, you get 46 chromosomes in the very first cell, right? That's called conception, essentially. And then within 20 minutes, this cell will divide into 2 new cells, which is crazy when you think about it has 6 billion letters now that have to be replicated exactly in this 20 minutes here and then created into 2 new cells. I don't know how that happens. I did the math on that once. It's like 50,000 new letters of DNA per second, and it has to be done correctly. If anything's wrong here in these first few divisions, the embryo will abort. And this is what happens in about 75% of pregnancies in North America, spontaneous abortion within 24, 48, 72 hours. And the reason why often is because there's just too much that's wrong here. Like there's miscalibration of all of these chromosomes trying to get together. It just doesn't work. So it just says, nope, gone. The woman doesn't even know that she was pregnant, essentially. So in fact, if we kept dividing at this pace, every 20 minutes, one cell becomes into 2, and 2 goes to 4, and so on. Obviously, it's exponential growth. By the time you were the age of 18, you would be as large as the sun. So obviously, that doesn't happen. This process slows down, and now what happens as you're getting older, your cells are still dividing, and they're still being made into 2 new cells, 1 cell into 2 new cells. And that happens about once every 24 hours. And then that doesn't even happen every 24 hours because cells have jobs. Like the cell in your nose or the cell in your pancreas is going to do its nose job or its pancreas job for a period of time. Then once it gets a message that says, hey, okay, you've done a good job for a week or a day or a few weeks, let's divide into 2 new cells. Then that takes about 24 hours. So let's keep going with this math here. This cell is then going to divide into 2 new cells. Remember that the DNA in these cells are identical to one another, and they're both identical to the original cell, and so on. So every one of your cells, if you were to take a cell from your pancreas or your nose, all have 46 chromosomes, all have the same DNA in them. So we can come back here. So my point is that in these first original 2 cells, the sperm and the egg from mom and dad, there were chromosomes, and there's 23 chromosomes, and we can label these chromosomes. We can say, oh, this is chromosome 1, and there's a chromosome 1 over here in the egg also, and there's a chromosome 5 over here. And the reason why we can do that is because they look the same, they're the same length, and they code for the same characteristics. Now, these are called genes that code for characteristics from chromosomes. They're just portions of the DNA. We have about 20,000 genes, which means we have about 20,000 characteristics. That's not true. We have about 20,000 genes which code through transcription. You learned about this in December. Through transcription, a message called mRNA is made. It then goes out to the cytoplasm. And then that message is translated into a sequence of amino acids, which is then folded appropriately into a certain protein. The reason why I paused there was because we have 20,000 genes, but we make over 8 million types of proteins. And so these things have to be folded in weird, mysterious ways. We'll get to that later, and then also we'll get to that complexity in AP biology, and I hope that you take that class. But it's the protein. I'm going to circle these guys over here. It's these guys that actually do the stuff, like create height in an organism, or tongue width, or hair color, or eye color, or anything like that. It's the protein that's important. Well, what I was saying was you have these chromosomes that code for the same characteristics, but it could be a little bit different. You know, mom could have a gene that creates a protein that creates blue eyes, and dad could have a gene that creates a protein that has brown eyes. And when they get together, you can look at these. You can look at these genes, and I'm just going to abbreviate those as a letter here. Let's say a big T and a small t. So remember what these are. These are sections. I don't know if you can see this anymore, but there are a bunch of nucleotides on the DNA, maybe let's say 1,000 nucleotides that we've abbreviated as a single letter, and they're really portions of a chromosome that then will be transcribed and translated to create a certain protein. So you could see a dominant or a recessive gene here, which is going to create a characteristic. And I know that you went over Punnett squares in middle school. Let's imagine, if we look at this picture, that mom has a gene that will create a protein that confers height or tallness to her. But also, let's say that she's heterozygous for this, which means hetero means different. And so she also has a gene that's going to confer a protein or create a protein that says short. But we know, so here's my little legend down here, we know that there's a dominant and a recessive gene, and the dominant gene is going to dominate over the recessive gene in the expression of that characteristic. So maybe we call her tall. Dad, on the other hand, in this instance, let's say that from his two parents, from his mom, he got a chromosome 10 in this first cell over here that had a little t, and from his dad, he got a chromosome 10 in this first cell over here that had a little t. And so let's put these together. These are called Punnett squares. You've probably done them before, and I know the kids like to do these. So thank you. Sorry about that. What these refer to, we're almost done here. What these refer to are different combinations of genes that the parents can give to the baby. And so in this first zygote here, so the woman has a big T or a little t that she can give to the baby, and the dad has a little t that he can give to the baby. And so what we see here is that there's 2 out of 4, or 1/2 of the gene combinations that will cause the baby to be what we call short due to proteins created that confer shortness, or there's 2 out of 4, or 1/2 of the gene combinations that can be created that will confer height to the baby. So this is a gene that exists on a chromosome that creates a protein that causes something to be a little bit taller. That's how this stuff is done. So go ahead and press pause. What I'd like you to do is on your piece of paper, go ahead and draw these things, and then I would like you to talk these concepts through to your favorite stuffed animal or your neighbor or whoever is around. I'd like you to push pause for about 3 or 4 minutes here and do this. Copy all this stuff down, please, and then talk.
Your body is made up of 50 trillion cells. Cells come in many different varieties with many different functions. But inside almost every cell is a nucleus containing 99.9% of your genes, and mitochondria containing a few more genes. All told, you have nearly 20,000 genes. Your genes are small parts of a long molecule called DNA, or deoxyribonucleic acid. If you lined up all of the DNA containing all of your genes, it would measure 6 feet long. But it's coiled so tightly that it fits in just one cell nucleus. DNA is a double-stranded molecule composed of sugar, phosphate, and 4 different bases: adenine, thymine, cytosine, and guanine. These bases spell out the language known as the genetic code. The number and order of these four bases determine, for example, whether you are a chimp, a cow, a banana, or a human. Most genes are recipes for making specific proteins. These recipes are passed down from parents to children, from generation to generation. When someone says, "You have your father's hair," what they mean is, you appear to have inherited a gene or genes from your father that makes a protein that instructs your hair follicle cells to produce hair that curls like your father's.
Please repeat this 2 or 3 times. This is true. And I feel bad for my daughter because this is the way that I would explain it to her. I feel bad for her. I feel nothing other than sorry that she was my daughter. She's nice. I think I'm nice, but this is what she had to live with forever. It's really sad.
But they usually opt for the shorter version. Genes tell a cell how to function and what traits to express. More specifically, gene regulators turn different genes on and off in different cells to control cell function.
This is kind of, I'm sorry to interrupt again. This is kind of what we're talking about in this unit right here. You see how there's a foot coming out of that guy's head. And the question is, can the cells in someone's scalp create tissue, create bone and toes and all these, a heel and ankle, all these things? And the answer is, of course, yes. So the question becomes, why do some cells express some genes and not others? All cells have all genes, but some of them are turned off. You don't want to create foot tissue on the top of your scalp. Or maybe you do if you want to do handstands all the time. But the point is that it doesn't happen normally. So just pay attention in the rest of the next few days, and I'm sure you'll get this concept down. It's my job to help you. We'll finish.
The long molecules of DNA containing your genes are organized into pieces called chromosomes. Different species have different numbers of chromosomes. Humans usually have 46 chromosomes, 2 sets of 23, or simply 23 pairs of chromosomes. Chimpanzees have 2 sets of 24, or 24 pairs of chromosomes. Rhesus monkeys have 21 pairs of chromosomes. Cows have 30 pairs of chromosomes. Chickens have 39 pairs. Fruit flies have 4 pairs. And bananas have 11 pairs. So, what percentage of the DNA in your chromosomes do you share with other species? You share 93% of your DNA with the rhesus monkey and 98.5% with our friend chimpanzee. How about with other humans? 99.5%. So,
I want you to understand this. If you were to pluck any 2 humans from this planet, take someone from Mongolia, north of China, and someone from Panama or Costa Rica, and you were to then analyze their letters on their DNA and all those 6 billion letters and just count them down, what he's saying is that 1 out of every 200 letters would be different. That's it. 199 out of 200 letters, exactly the same. Any 2 people on the face of this planet. And that is enough, just the 1 out of 200 letters difference, to make someone be so different on this planet. I just think it's amazing to me that so little information causes such great differences amongst people. Or you could look at it the opposite way. You could say, look, we're all alike. Maybe we should have social equity and help our fellow brother and sister. I mean, if there's anything that we've learned recently is that we are only as good as our neighbor or as healthy as our community. We need to look out for each other, and maybe this suggests how similar we actually all are. So I'll get off my soapbox. Today, what we're going to be doing is we're going to be doing this, a programmed introduction to genetics. There are 19 questions here. There's a thing on the back called the hybrid cross problem set. I do not want you to do this yet. We'll do it later. There's just 19 questions here. I'd like you to do them on a separate piece of paper. I'm going to show you. I've got that in the frame there. Question 17 just asks you to define 6 words and then give an example, a visual of each. This is question 17 in the packet. Just try to work on the packet. Here's what I don't know is how I'm going to assign this to you. Are we going to do it together in class? Are you going to do it as homework? I don't know yet because it's November. It's early December. So the point is I'll figure all that out when I talk to you. On your blank piece of paper, it should look like this, a program introduction to genetics on the top. Put your name, put the date, and then just start answering the questions. And then eventually, this is what you will turn in, this blank piece of paper that you filled out all these answers to 1 through 19 on. You're then going to staple it to the back of this piece of paper, and you're going to write a program introduction to genetics on the first line here. Because it looks like we're going to do a few more assignments as well to try to master this topic. Thanks for listening. Hope you're doing well.